So now going back to agrogenomics and pests genomics, our next speaker is Scott Geib from the USDA in Hilo, Hawaii, and he'll tell us about CiFi.
All right, I'll test you guys out. Aloha. That's all right. Okay. So I'm Scott. I'm from Hilo, Hawaii. We work in arthropod genomics. I'm going to talk a little bit about how we're doing a lot of sequencing on a lot of these Earth biogenome projects and how we can add in some more layers to some of the sequencing we're doing to create what I would call more holistic genome sequencing, and also present some new technologies such as this low input, high C sequencing. So predominantly what we do in our lab is what's called the Ag 100 Pest Initiative and the Genome 100. These are focused on agricultural pest insects and beneficials. So it says the top 100, but if you're out there and you have a bee or a pest that you want sequenced, you could probably send us our way, and we would sequence it. We're also working on the Genome 100, which aims to sequence not honeybee, but native bee pollinators in North America. So the Genome 100 project now has sequenced over 230 species. So again, these numbers really don't mean too much. The take home here, though, is a lot of these are one-off specimens. So there's something coming from overseas, if it's a pest insect, or there's some unique novel species of bee that's very hard to collect. So often many of our samples are rare, novel, and maybe represented by a single specimen, either for the species or maybe for the strain or population. So what we're trying to understand is how we can get as much information and data from that single individual insect. So this is just to show you an overview of some of the Ag 100 pest projects that we do. This is focused specifically on USDA-ARS labs across the nation, the different pest species that we've worked with. But the take home is that this generates a lot of times maybe just a genome assembly, things like the desert locust where there's projects ongoing, the spotted lanternfly. But sometimes there's also specific users with specific questions. So maybe I'm interested in evolution systematics. That has a very applied application in USDA and diagnostics of invasive species. It could be in functional genomics. There could be a researcher chomping at the bit at understanding insecticide resistance or some phenotype. It could be microbial ecology and multitrophic interactions, so things where people are trying to understand different layers of the insect. So if we just supply them with the reference genome, sometimes those labs don't have the capability to kind of take it further so that they can get to the point where maybe they're doing functional genomic experiments or whatever they want to do with their genome. So we're trying to look at how we can generate more comprehensive data sets that really sets them up for success. On the Genome side, that's our little Genome. You can say hi to him. You have what we think of as the conservation ones, right? The bees that are declining and all that kind of thing. But there are bee researchers as well that are doing very functional genomics type of research and want to know more about their bee maybe than just the genome itself. So here in Hilo, we have our Hungry sequencer. Those are googly eyes that we've placed on our Revio. We can sequence about 4 plant or insect-sized genomes a day. And we've budgeted about $1,000 per genome. That's very conservative, and the pesky people in the biotech industry keep making these things cheaper. So now we want to see how we can do more things with the $1,000 that we've kind of set aside for each individual insect. So mid-December, they talked about the Spark chemistry coming out as well in a SmartLink update. So we also want to look at how that's changing maybe the amount of output we can get and maybe what we can do with one flow cell. So I'm going to tell you a little bit about a Spark run we did literally last week. This is a completely random specimen. This is a ground nesting bee, part of the Genome 100 project. It has a 375 megabase genome size. So if we want to really kill it with coverage, we could generate 37 gigs of data, and that would be 100x coverage of the genome, way more than what we actually need. So they said we could put less DNA in and get more yield out. So that's exactly what we did. So we're loading about 50 nanograms of bound library. This is more of a number that somebody who runs a sequencer will understand. This isn't the amount of input going into the library prep, but the amount being loaded on the sequencer. This is much less than what we were loading before. We ran it last week. The data looks pretty good. We got a pretty good read-linked distribution and very nice quality metrics. And this generated over or exactly 120 gigs of data. So when they say 120, you're getting your 120. That being said, though, 93 gigs of this data was over 15 KB in length, which is a real boost for us as well. We have a second run going on right now. The problem here is they're also generating a ridiculous amount of data. So this is other bees with other small genomes that have over 100 gigs of genome or of data being generated for them. So now we're getting 4 times too much data to still be over-sequencing our genome by 3 times the amount we actually need to. So we really want to look at how we can utilize the rest of the flow cell to get a more complete or comprehensive genome coming out of it. So I create this concept of holistic genomics from a single insect. Things like generating your genome sequence so you can make a nice assembly and make your snail plot and feel good. Generate HiC data to understand your genome in a chromosomal context. Look at the microbial associations of your genome. So look at things like gut complexity. I mean, the talk before kind of set that up very well. Also look at things like gene structure and content. And then also regulatory architecture and epigenetics. So I think we're pretty familiar with genome sequencing, HiC sequencing, microbes, and gene annotation. I'll go a little bit into what PacBio can do on the regulatory architecture side, though. So first, it is a 5-base sequencer and can generate CpG-style 5-methyl-C methylation calls. We don't see that a lot in our insect species, but we can also introduce artificial DNA modifications during the library prep process. So one of these, and Jake mentioned this a little bit in his beginning, is this fiber-seq. So this is inferring regulatory elements or open nucleosomes in the genome. I'm not going to go too much into this because he already did it earlier. But basically, you do this while you're extracting your DNA. So you sequence it, and you're not having to pay any extra money or do anything extra. You just get this data, and it's great. He also, this is Andrew Sturgakis, who kind of developed the fiber-seq protocol. He also introduced this routine multi-omics concept in human genetics. So by doing the fiber-seq reaction up front of your genomic DNA, do SMRT-Bell sequencing, and pair that with cDNA sequencing. He says mass-seq here, but this would be kinnex. You can generate from one cell, basically your whole genome, your epigenome, your methylome, if you have that, and a transcriptome, and sequence that all in one run. That's great. We want to look at how we can do that from a single fly, though. So we're going to do a little scissor action. We don't really cut them with scissors. We usually break them in a cryogenic state. But we start with his fiber-seq modifications, with which we can do on insect tissue pretty easily, and generate our genomic DNA in SMRT-Bell runs. I think the next talk will talk a little about how you can also co-extract RNA at the same time as your genomic DNA extraction and do cDNA sequencing and pull those libraries. We're also interested in how we can integrate then things like 16S amplicon sequencing or metagenomic profiling, as well as the HiC component. So this would be formaldehyde fixing your flies. So this is where this little part of the fly has to be separate. He doesn't have to be half a fly necessarily, but he is getting a different treatment than some of the other tissue. So you do your formaldehyde fix, you basically are fixing at the nuclear level and then a HiC prep. And then can we conceptually pool this all into one run and sequence it and get all this information from a single fly? So this is what Andrew kind of had before, the genome, the methylome, the epigenome, and the transcriptome. And we're just trying to add in HiC architecture and microbial associations. But most people would say, isn't HiC on PacBio science fiction? And as we go to the next slide, so Megan's lab at UC Davis has developed this CiFi sequencing protocol call. So this is HiFi-based sequencing or HiC sequencing on a HiFi system. And so I'll tell you a little bit how it's different than your standard HiC protocol. So we're still cross-linking our cells and we're digesting them. In our case, we tried with Hindi3 and DPN2. We do not do a biotin fill-in, and there's no enrichment of the libraries. This is what you would call classic 3C library, and not really a HiC library. We then reverse the cross-link and do DNA purification. So the DNA you're purifying here, can I point? I can point, look at that, I can point. So you see these different colors, so these actually are different parts of the genome that were proximity ligated together. So you're creating a concatenation of segments that are now created into a long read. Normally in a HiC library, you would fragment at this point and try to just capture the two paired reads. What we're gonna do instead is sequence through that whole thing and create a lot of combinations of that. The downside is, so we don't do any shearing, we're sequencing long molecules containing lots of proximity ligations. The downside is you've already formaldehyde fixed this DNA, you've already digested it, and then you had to ligate it back together, and then you had to undo the cross-linking. And all of that messing with the DNA makes it really recalcitrant to sequencing at a single molecule level, both on PacBio and Oxford Nanopore. So what we've introduced is a PCR amplification. This isn't as much to amplify the library and make more of it, but rather to make fresh, high-quality template that's amenable to sequencing. And then we go through smart bubble adapter ligation and PacBio sequencing. So Megan's group has done this on human cell lines. They show that it maps just as good across the human genome. The one thing I want to point out is you're talking about number of segments in this read, and then the length of those segments within the read, right? And so depending on the restriction enzyme you can pick to get fewer segments, but longer reads, or longer segments, or more segments and shorter segments, right? So do you want to cover the genome more, or do you want a longer read that might map uniquely into the genome better? This is what a single CiFi read will look like. So you can see this example one's about 10 kb long, and you can see all of these segments mapping across the chromosome 2 in the human genome. They also show that it does map better in difficult to map repetitive regions because the read is longer. You're not mapping a 50 base pair Illumina read, you're now mapping a 2 kb PacBio HiFi read. So then Shenna Sim and Renee in our lab, as well as myself, adopted this to being used in low input insect tissue. So this is going back to our scissors and looking at this formaldehyde fixed side. So there's a picture of a fly here, but we care about other insects too. So we tested this in a moth, a mealworm, which is a beetle, and then 2 fly species. And we followed pretty much the protocol that Megan has put together and generated this nice library that's about 6 kb in length. All of these were pooled onto one flow cell, and we're generating about 1 million HiFi reads for each, with that fragment length between 6 and 8 kb. That being said, I really have this concept in my mind of pooling this with a HiFi library, like a standard whole genome sequencing library. So if you see the tail here, can we size select about there and focus on the tail and create a sequencing library that's now going to be the same size as our DNA sequencing library, right? So we did that and we sequenced it. We got a little double hump, but it's pretty good. It has I think this was some of the tail coming down from the other side, but it now, when we sequence the exact same pool of individuals, we're hitting these more HiFi read lengths that are above 10 kb long and that are on par with regular PacBio whole genome sequencing. So I'm going to take a break right now. I'm going to come back to that. But all the examples I'm going to show are in our tip-rooted fruit fly examples here. So we work out of Hawaii because these flies were introduced to Hawaii back 100 and plus years ago, and they're worldwide global pests. There are different species that we work with. They're all pretty distinct in their distribution, but all of them are polyphagous and target a lot of host fruit. So when you go through the airport, we're the reason why you can't bring the apple, or you gotta go through X-ray, or if you're going back from Hawaii, you have to fill out a little form that says, I promise I'm not bringing mangoes or something like that. Most of the control for this is using the sterile insect technique. So this is inferred through a translocation on the autosome and the Y chromosome to put phenotypic traits such as pupil color or temperature-sensitive lethal onto the Y chromosome to make it sex-linked. These flies are then reared at high numbers, males are collected, they're radiated, and then released by the millions or billions in Southern California, maybe around here. Maybe if you look up, you might see some flies falling out of the sky. So I'm going to focus on different ways we've used PacBio sequencing in characterizing this system and working towards improving it, including using the HiC data for characterizing structural variations in this translocation, as well as using long HiFi reads for characterizing the pupil color morph. Also, gamma radiation is bad. It makes your flies sick, basically. So we're interested in other genes that are regulating sex determination and how we can control this molecularly so that we don't have to induce sterility through irradiation. And then also when we release these flies, we want them to be happy and healthy. So we're interested in their microbiomes and how we can improve their microbiomes for their fly health. So getting back to the HiFi CiFi story, we took a Mediterranean fruit fly and cut them in half. Half of them is whole genome sequencing library, half of them is a CiFi library, and sequenced on a smart bell at about a 1-to-4 ratio. So 20% of the reads going from CiFi and 80% for HiFi. He gets fed to our hungry sequencer and generated a read distribution that looks pretty good for a HiFi library. Because we pooled two things, you see a little bit of spikiness here, but the breakup of this was about 1 million or a little less CiFi reads, and then the rest being HiFi. So this is one cell, CiFi, HiFi together, and we're going to put this together into an assembly. So like I said, this is a segmented read, a read with a lot of segments in it, it's going across. And so the first thing we use is the epitome POR-C pipeline. So POR-C is the nanopore-based HiC sequencing pipeline. These CiFi reads look exactly the same as them, except in our case, they're much, much longer, and they're of HiFi quality. So we do modify the mini-map mapping parameters to be for HiFi mapping data. Otherwise, everything is the same. So you get a bed file of all the contact pairs. And then, like I was saying, you can decide if you want more segments or if you want more length. So now with our super long reads that are now 10 kb and longer, we're getting as many as 200 pairs. So 200 segments. No, we're getting 20 segments, but if you do the math, that creates 200 pairs here. And with like more common or the less common cutter, we're getting maybe 5 to 10 segments, which is creating less pairs. But some of these read lengths can be very, very long, going out to even longer than 8 kb or segment lengths. So you can choose your own adventure, what you make sense in your system if you want the longer reads or more connections. But we've used this now to assemble genomes at a haplotype resolved scale. So we're just using HiFi ASM, and we use the CiFi read pair data as HiC data up front using the dual scaff option to resolve 2 quality haplotype assemblies. The CiFi BAM output of POR-C can be directly fed into yet another HiC scaffolder or gas to facilitate scaffolding of each haplotype, and then we can do things like explore sex-linked translocations, like we know there is one in this line. So you can see going across, you can see this is this chromosome 1, 2 times haplotype 1, haplotype 2, chromosome 2, haplotype 1, haplotype 2, etc. going through. Just a way of showing that we've resolved both phases. I want to say too that CiFi coverage is now reaching into regions of the genome previously not captured at low read depth. So this is a HiC assembly of a region, and you can see it really drop off in the centromere region. And this is CiFi, and you can see the CiFi reads kind of working their way through the centromere, which is pretty awesome. So we're getting better coverage of these heterochromatic spaces. You also apparently need very low numbers of reads, especially when you have a HiFi assembly to put the genome together. So this is just L90, so we want a very low value that's equal to the number of chromosomes in our genome. And we continually keep downsampling pairs of proximity ligations. And we get down to about 250,000 pairs, and that's where it starts to fall apart and we're not able to generate a chromosome-scale assembly. These 250,000 pairs were from only 70,000 HiFi reads because of the desegmentation that happens. So you could potentially spike in a very tiny amount of HiC data or CiFi data into your library and get enough data for assembly. So we can see now that this translocation actually, this is shown as like a tip of the autosome is carrying the Y chromosome, but no, it's not. It's internal to the autosome, and you can see the Y chromosome being nested here within the autosome. We also can look at the placement of the autosome within the Y chromosome. You can see it on this haplotype that doesn't contain the translocation, and the matching to the Y chromosome now containing a large chunk of the autosome. So this is what's driving the sex linkage of these traits in the fly. I'm going to jump over now to understand the pupil color that's happening. So we've been working for a long time of trying to characterize ebony as the causative trait for black pupae in this line. And using all this other technology, it's kind of like a little bit granular, but when we add in the HiFi reads, this is a heterozygous male and then a homozygous female for the deletion, we can just see it characterizing this large, it's about 23 kb region that basically is excised ebony from that completely. So it's not that the gene is disrupted, it's that gene just disappeared completely, and that's what's creating the phenotype. We then use CRISPR and can recreate those phenotypes in 6 species. We had a group in Queensland work with us on a species that we don't house internally. We're also interested in how we can use isoseq and epigenetics to identify sex determination factors. So people have characterized this maleness on the Y, orchestrating sex determination for males on the Y chromosome. So if you knock it out with double-stranded RNA, you get female-like males. 2 minutes? All right, perfect. But this study was done in the absence of a resolved Y chromosome. So they just know this thing works. If you knock it out, it stops working. The problem is when we have a resolved Y chromosome, there are 6 copies of maleness on the Y chromosome. A lot of them aren't really genes. There's all these transposable elements around. Things are jumping around and all that kind of thing. RNA-seq kind of just maps generically across. When we try to predict these genes, the gene isn't predicted completely. But when we add in the isoseq data, we can see very clearly the active MOI here. There's a whole larger region of this having no mapping of the other 5 copies. But this is the real MOI. So now if we want to fiddle with it to create male-only lines or female-only lines or those kinds of things, we can. We can then add in FiberSeq to look at the epigenetic characters that are regulating it, and you can see these little fire elements upstream of genes on the Y chromosome. We're also interested in making our flies happy and healthy. So we utilize full-length 16S sequencing, very much like the talk before. What we're trying to do is give probiotics to the flies so that they're more happy, healthy when we release them. We're trying, those probiotics are supposed to be very similar strains to ones that are already harbored in the gut. So it's very critical for us to have a full-length 16S sequence so that we can discriminate between the strain that we're putting into the gut and these strains that are already there. This is mostly at a species level or subspecies level for these strains. And this seems to work very well. We basically, right when these flies emerge as adults, they have a gut that's empty, just waiting to be filled with bacteria, and that's a great time for us to put in this Enterobacter species that can improve their gut health. So the caveats and takeaways from a single fly, you can do a lot of things. Some of it's for free, like the de novo assembly, 5-methyl-C, fire determination. You can couple it with isospore sequencing to get a comprehensive view. You can now generate high C data using the CiFi protocol. Microbial associations can be characterized robustly on only a fraction of the cell and sample, but that being said, you need to have high quality samples, right? So a lot of this is being done from intact nuclei and things like that. So you need to have extremely high quality ability to isolate nuclei, and you're only getting the epigenetic signatures, the transcriptomic signatures, microbial signatures of the time points and tissues that you're sampling. But in concept, you could do all this on a smart cell. You could pick and choose of what you want to do. But I think with the outputs now and with the size of a lot of our genomes, we have the ability to get a little bit more out of the sequencing we're doing on a single smart cell. So with that, this is our lab group in Hilo. And thank you so much for the invitation.